You know, sometimes science really does feel like a great detective story. You've got a mystery, a confusing set of clues, and then a brilliant technique that just cracks the whole case wide open. And the story we're diving into today is a fantastic one, a biological whodunit that takes us deep, deep inside our own cells to figure out how a perfectly good gene can suddenly go bad. So, where does our story start? It all kicks off with a single, really baffling observation inside a breast cancer cell. Scientists were looking at this one crucial gene, a gene that's supposed to be our protector. But in this specific cell, it had just gone completely silent. And the big mystery was, why? And this is really the heart of the matter, isn't it? We're not talking about a gene that's broken or has a typo in its code. No, this is about a perfectly healthy gene that has been mysteriously switched off, which then lets a cell grow totally out of control.
Okay, so the gene at the center of this whole mystery is called CDKN2A, but let's just call it P16. It's a lot easier. Now, think of P16 as a guardian, or maybe like the brake pedal for cell division. Its entire job is to step on the brakes and stop cells from multiplying when they shouldn't be. To put it simply, you really want this gene to be working.
And here's the twist in our story. When the researchers looked at the actual DNA of this cancer cell, the P16 gene was perfect. The code, the A's, T's, C's, and G's, it was all there, exactly as it should be. The gene was present and accounted for, but it just wasn't doing its job. The brake pedal was installed in the car, but something was holding it up, preventing it from ever being pressed.
So, to solve this mystery, we have to look beyond the DNA sequence itself. We've got to enter this totally fascinating world of hidden instructions, a layer of control that sits on top of our genes.
And that brings us to the amazing world of epigenetics. The best way to think about it is like this. Imagine your DNA is a giant cookbook with all the recipes to make you. Well, epigenetics is like all the sticky notes, the highlights, and the notes scribbled in the margins. These notes don't change the words in the recipes, but they absolutely change which recipes get cooked and which ones get ignored.
So how does this actually work on a physical level? Well, your DNA isn't just floating around. It's actually spooled incredibly tightly around these little proteins called histones. This whole DNA and protein package is called chromatin, and those sticky notes we were just talking about, they're actually tiny chemical marks that get placed right on these histones, and they act like little on-off switches for the genes wrapped around.
And believe it or not, we've actually cracked the code. Scientists have figured out what these different marks mean. Some marks, like H3K9AC, are basically a big green light. They shout, "Gene on, get to work." But another mark, like H3K27ME3, and remember that one, is a giant red stop sign. It screams, "Gene off, stay silent." It's this very specific code that holds the key to our whole P16 mystery. Okay, so this hidden code exists all over our DNA. But how on earth do scientists actually read it? I mean, you need a pretty specialized tool to scan the entire genome and find out exactly where all these on and off switches are, right?
Well, that tool is called CHIP. That's C-H-I-P. And you can think of it as the detective's special magnifying glass for epigenetics. It lets researchers zoom in and see exactly which of those histone marks are sitting on any given gene across our entire genome. It's an incredible piece of technology.
So with a technique like CHIP, scientists can create a detailed map of all these epigenetic switches. They can see which genes are being activated, which are being shut down, and, most importantly for our story, how this whole pattern gets messed up in diseases like cancer. And of course, that might just reveal brand new ways to fight back.
All right, so we've got our detective's tool, CHIP. We know what it does, but how does this powerful technique actually work? Let's take a quick peek inside the lab and walk through the process, step by step.
Yeah, I know this workflow might look a little intimidating at first glance, but it's really just a forensic investigation happening at a molecular level. And we can break it down into a few really key steps.
Okay, here's how it goes. First, you add formaldehyde to freeze everything in place. It's literally like taking a snapshot of the crime scene. Then, you use sound waves to break the DNA into smaller, manageable bits. Now for the magic step. You use the special antibody that acts like a tiny, super-specific magnet to pull out only the histone mark you're looking for, say that gene-off signal. And finally, you just see what DNA was stuck to it. Whatever DNA you find, well, that's the gene that was being silenced.
All right, now we get it. We understand epigenetics, we understand the SIP technique, so we can finally go back to our breast cancer cell and solve the case of the silent guardian gene once and for all.
So, the researchers fired up the CHIP machine with one very specific question in mind. What epigenetic marks are sitting on top of the P16 gene in these cancer cells, and how does that compare to healthy cells? They were hunting for the sticky notes that were telling this crucial gene to just stay quiet.
And the results? Well, they were absolutely crystal clear. In the healthy cells, the P16 gene had tons of the on signal and very little of the off signal. It was active, just like it should be. But in the cancer cells, it was the complete opposite. The on signal had vanished, and the gene was just covered in that repressive off mark we talked about earlier, H3K27ME3.
And there it is. Case closed. The mystery is solved. The gene itself wasn't broken or mutated at all. The cancer cell had simply rewritten the epigenetic notes, placing a powerful off switch right on top of this critical guardian. And by doing that, it completely disabled the cell's safety brake.
But you know, this discovery is so much bigger than just one gene or even just one type of cancer. It completely opens up a new way of thinking about disease and how we might one day be able to treat it.
Using tools like CHIP, we're now finding these kinds of epigenetic miswrites in a huge range of human diseases. It's a fundamental biological mechanism that can help explain why some people get sick and others don't, even when they have very similar genetics.
Now, it's important to be real here. While CHIP is incredibly powerful, it's not perfect. It can be a really difficult and time-consuming process, and you need a lot of starting material. We're talking millions of cells. Science is always working on making its tools better, but for now, CHIP is still the gold standard for asking these kinds of questions.
And that brings us to the most incredible final thought. Remember, that P16 gene wasn't permanently broken. It was just silenced by a removable chemical tag. So this leads to maybe the most exciting question in all of medicine today. If we can read this epigenetic code, can we develop drugs to erase the off marks and write in new on marks? Could we reawaken our body's own guardian genes to fight disease from the inside? The power to not just read, but to rewrite this code, well, that could change absolutely everything.